In Section 5, we'll be exploring weed reproduction by seeds. As indicated here in the overview statement, weeds rely on seeds to disperse their species over space and time, and that ensures their long-term survival. So in this section, we'll be looking at weed reproductive strategies, self versus cross-pollination, for example, weed seed dormancy types, both physiological, physical, and other types of seed dormancy. And at the end, we'll take a look at some of the many ways that weed seeds get dispersed. After finishing this section, you should be able to define seed viability and seed dormancy, explain what Baker's rule is and some of its advantages, describe the physical, morphological, and physiological dormancy mechanisms of seeds, and explain the role of phytochrome in light control of dormant seeds, and then be able to describe 6 mechanisms of weed seed dispersal and examples of different dispersal symbioses. As stated in the beginning, the primary role of seeds is to disperse the species over space and time. And in doing so, that ensures the long-term survival of the species. Okay. A botanist from University of California system back in the 1960s kind of was interested in weeds and took kind of a big overview of the world's weeds and came up with what he referred to, or what some people referred to as Baker's rule in 1967. The so-called rule, it's not really a rule, but that's what it's called, is that plants capable of uniparental reproduction, that is self-pollination, are more likely to be successful colonists, weeds in other words, than plants that are self-incompatible, those that have to cross-pollinate. Well, there are definitely some survival advantages associated with that Baker's rule strategy of being primarily a self-pollinated plant. One is that a single plant invader can give rise to a new colony. It doesn't require the presence of another species to provide pollen to its flowers or anything like that. It's self-contained and can carry on reproduction all by itself. The second advantage is that the offspring produced from a self-fertilization event are just as genetically fit as the mother plant. After many cycles of inbreeding, essentially a plant becomes homozygous and its offspring are basically genetically identical to the parent plants, so they should be just as genetically fit to survive and reproduce as the parental plant. Thirdly, then, a low but steady frequency of outcrossing ensures the incorporation of new genes that allow it to adapt and evolve. So these so-called Baker's rule plants are not exclusively self-pollinated plants. That's very important to know. They do carry on a low but infrequent rate of cross-pollination. Usually that's in response to some environmental stress. So these so-called Baker rule weeds are weeds that may self-pollinate 90-95% of the time or more, but occasionally, in response to certain situations, they are capable of outcrossing, accepting pollen from a different plant. That allows the incorporation of new genes to allow it to continue to evolve and change over time. Well, a couple of definitions here for clarification. A viable seed, when we talk about a viable seed, we're really just talking about a seed that is alive. And the way we know it's alive is that it's carrying on respiration. It may be at a very low rate if the seed is dormant, almost imperceptible, but living organisms, aerobic organisms like seeds, have to consume oxygen. And there are biochemical tests we can do to show that. A dormant seed, on the other hand, can be defined as a viable seed that does not germinate even when conditions are favorable. For germination. So this is saying that you give a seed adequate temperature, moisture, water, everything it needs to germinate, and still won't germinate. As long as the seed's viable, then it can be defined as a dormant seed. Well, there are three major categories of seed dormancy: morphological, physical, and physiological. And it's important to know that some plants may produce seeds that have a combination of different dormancy types. Some plants may have a combination of morphological and physiological dormancy, physical and morphological, and so on. So it's not confined to any single type necessarily. Now, in terms of evolution, these are ranked in order from oldest to newest on the evolutionary scale. So plants that have morphological seed dormancy have been around a lot longer than the more highly evolved plants that have physiological seed dormancy. So physiological seed dormancy represents the most advanced form of dormancy on the evolutionary scale. Let's take a closer look at these different types of dormancy. Morphological dormancy is the oldest and most primitive form of seed dormancy, and it's caused by a small, underdeveloped seed embryo. That is to say, when these seeds are produced by a mother plant, they're not fully ripe or mature. They contain this underdeveloped seed embryo that usually requires another period of a few weeks before it reaches a stage of maturity so that the seeds can germinate. So you'll find that in species like poison hemlock and the lulu palm, where the seeds, once produced, will remain dormant for a period usually of several weeks, even after they're released from the mother plant. This is not a very common dormancy mechanism in weeds, the exception being poison hemlock, but it is a fairly rare type of seed dormancy that only occurs on a few known weedy species. Physical dormancy, as the name implies, is caused purely by a physical barrier, and that physical barrier is the water impermeable layers of the seed coat or fruit coat, which prevent the penetration of water. So it basically keeps seeds dormant by keeping them dry. Dormancy is terminated whenever the seed coat or impermeable layer gets ruptured by a process called scarification or by natural things like weathering, microbial degradation, etc. Scarification is just any treatment you can do to a seed that ruptures that seed or fruit coat. It can be using sandpaper or a file or putting them in through a physical process that somehow causes abrasion on that seed coat, just enough to allow water to penetrate. So some very common weed seeds have physical dormancy: velvet leaf, Jimson weed, tall morning glory. And if you look at those seeds, you'll see that the seed coat appears and is in fact very durable and tough, and they will sometimes stay intact for many years before that seed coat is abraded enough to allow water to penetrate. Finally, physiological dormancy is the most advanced form of dormancy on the evolutionary scale, and it's caused by physiological processes inside the seed that are often controlled by the interaction of plant hormones and environmental factors. Depending on the species, dormancy in seeds with physiological dormancy can be terminated by stratification, which is exposure to a prolonged cool, moist period. So this would be primarily in soil we're talking about, unless it's artificial conditions. So, for example, we might be talking something like 6 to 8 weeks in a moist soil at temperatures somewhere between 30 and 50 degrees Fahrenheit would be a good stratification temperature that would induce non-dormancy in dormant seed. Hormones are also involved in physiological dormancy. There are some hormones that promote dormancy, like abscisic acid. Whereas others tend to release dormancy. Gibberellic acid will release dormancy in many species. Ethylene can also break dormancy in some seeds. Now, after ripening is just a process that requires a period of warm, dry temperatures. They promote maturity and will break seed dormancy. The next one, light, is required to break dormancy in some species. It tends to be most operative in small seeded plants that germinate at or on the soil surface. So we'll talk about those a little more in a minute. So if you look at these different types of physiological dormancy, different plants have these. Stratification, the requirement for stratification to break dormancy is very common in summer annual species. If you think about that, now these are seeds that require a prolonged cool, moist period. In other words, over fall and winter in order to germinate. So stratification plays a large role in why summer annuals behave as summer annuals, why they have a summer annual life cycle. It's because they don't typically germinate until after they've received this stratification treatment. So they're primed and ready, non-dormant, ready to germinate in the spring when summer annuals normally germinate. After ripening, in contrast, is a physiological seed dormancy process that you'll often find in winter annuals. And there again, think about how winter annuals operate. They germinate in the fall and or the spring, depending on if they're obligate or facultative winter annuals. So those are primed to germinate after they've sat on the soil surface all summer, soaking up those warm, dry conditions that allow after ripening to occur. So they're primed and ready to germinate, and they become non-dormant in the fall. For light-sensitive seeds, again, this occurs mainly in annual species that have very small seeds that have to germinate right on or very near the soil surface. So let's take a closer look at this light requirement. Arguably one of the most advanced types of physiological dormancy. So this light requirement for germination is, in fact, well suited for small seeded weeds. If you think about where seeds germinate in the soil profile, weeds that have very small seeds have to germinate at or near the soil surface because they have a limited amount of reserves to push that seedling above the soil surface and then start carrying on photosynthesis. So if small seeds are buried too deep, they either don't germinate, or if they do germinate, they die before they can emerge if they're buried too deeply. So these small seeds need a way of sensing if they're close enough to the soil surface to germinate, become established quickly, and start carrying on photosynthesis. So this light requirement for germination is very well suited for those small-seeded species. So this operates with the help of some specialized pigments found in plants called phytochromes. And a phytochrome is basically a pigment that acts like a switch to turn things on and off. When you shine red light on this pigment, it turns things on, like germination processes. When that same pigment receives far red light, though, it inactivates those same processes. In other words, seeds stay dormant if they only see far red light. So red light is this part of the spectrum here, kind of a dark fire engine red, and far red light is out here for a longer wavelength where it tends to get a darker or maroon type of color. So these phytochromes that I mentioned are groups of pigment proteins in plants involved in initiating these light-dependent growth processes, such as germination, but also it's involved in growth and flowering responses as well. So this pigment, as shown here, the chemical structure shown here, at least the business end of it, shows that when it's exposed to red light here, then it flips in the other direction, and now it kind of curves in this direction. And in this form, it can absorb far red light, and that will flip it back the other direction. So it acts as a chemical switch where red light causes it to flip this way, far red light causes it to flip back in the original conformation. So it's called a photoreversible pigment. And the key thing to remember is that red light always activates physiological processes with phytochrome, and far red light inactivates or deactivates those physiological processes. These two forms of phytochrome are called the PR and the PFR form. PR form just means it's the red absorbing form. And so it would be the inactive form of phytochrome you see here. It's not activated until it absorbs red light. Then it becomes the PFR form, which is now the active form that's going to stimulate germination responses in the case of seeds. This PFR form here, the active form, it can revert. If placed back in darkness, it will revert back to the inactive form. So overnight, for example, it's possible for that activated phytochrome to become inactive again as it goes through this darkness reversion. All right, so red light is light of about 670 nanometer wavelength, again this bright red color, and then far red light is out at 730 nanometers, longer wavelength, darker color. So full sunlight contains all the wavelengths, all the visible wavelengths detectable by the human eye. So when it's all mixed together, it appears like white light. Leaves appear green to us because the chlorophyll in leaves reflects green light. It does not absorb it, and so we see the reflected green light as a result of the chlorophyll. Chlorophyll absorbs the red and the blue wavelengths, so we don't see those because it's being absorbed. All right, out in full sunlight consists of a ratio of red to far red light of about 11 to 10. So almost equal amounts of red and far red light, a little bit heavier on the red side. So that means that under full sunlight, light-sensitive seeds that have a red light requirement for germination will get all the red light they need, the phytochrome will be activated, and the seeds will go ahead and germinate then in a non-dormant condition. One of the other things that chlorophyll does, though, in green leaves, because it absorbs red and blue wavelengths, it filters out a lot of that red light that it gets in full sun. So underneath a canopy of green leaves, you actually only get a ratio of about 2 parts red to 10 parts far red, because chlorophyll doesn't have any effect on the far red. It lets it pass through, as shown here. So the seeds that are on the soil surface beneath a canopy of green leaves are getting exposed primarily to far red light, and as a result, the phytochrome is inactivated, and the seeds will stay dormant. They won't germinate as long as they're underneath that canopy of green leaves. So we use that principle in talking to growers about the importance of getting early canopy development in their crops, in row crops like corn or soybean. Because once you shade the soil surface, you're actually preventing a lot of new weeds from germinating because they're not getting the red light they need. They're being bathed in far red light passing through the canopy, and that keeps the phytochrome inactive and prevents germination.